Okay, so let's think about enzymes here. And it's interesting because enzymes are a class of proteins, and we just talked about proteins and the way that they function, the way that they can be denatured with excess heat, excess temperature, etc. And so now we're going to kind of zoom in and think about how their roles are, but we want to remind ourselves that they are proteins, right? And so the properties that govern proteins will also apply to enzymes. Now, what is the overall function of an enzyme? It's supposed to be a catalyst for reactions, especially within our physiology. So it's going to speed up the rate of the chemical reactions that occur in our body. They will essentially decrease the activation energy, and the activation energy is the barrier for that reaction to occur. So if we decrease the barrier, we make it more favorable for the reaction to proceed, but we also just increase the efficiency of the reaction, the amount of time that it takes, the amount of resources that's required for that reaction. Enzymes are going to be able to bind to the reactant, which is also called a substrate, which is shown here in this equation at the bottom. It will bind to the substrate, and then via interaction with the enzyme substrate complex, that is, we're going to end up with a product and the enzyme on the other end of our equation. We also want to remind ourselves that these bidirectional arrows means that this can proceed in both directions. So we can bind enzyme to substrate and end up with a product. We can bind product to enzyme and end up with a substrate. So the reaction can proceed forward and the backward direction as well. Now, here's an illustration that shows us the way that enzymes decrease the reactivation energy. And we're comparing in the red curve what the reaction would look like in terms of the energy required on Y and the reaction coordinate. This can also be described as time, right? How the reaction proceeds. So with regard to those two things, the curve in the red is what happens in the absence of an enzyme. Notice that it takes much more time. The steep curve here is not time, excuse me, much more energy. So the steep curve here reflects the activation energy without the enzyme. And it takes a lot more energy for this reaction to proceed in the absence of the enzyme. Now, in the blue is the reaction proceeding with the enzyme present. So we have significantly lowered the activation energy, right? Almost half is required. So this is a much more efficient way for the reaction to proceed. It takes less energy. And then we also are going to generate the products in a slightly shorter amount of time, as we can see, right? So the reactants here, in this example, it's carbon dioxide and water. And the products here is carbonic acid, right? Carbonic anhydrase is the example of the enzyme, although it's not shown here, that is needed for this reaction to proceed. So if we didn't have carbonic anhydrase, this would take a lot more energy. And in the presence of carbonic anhydrase, we can make carbonic acid with much less energy required. Okay. Now let's think about the theories that explain how enzymes work. We have two different theories, and I say theories because these have been described by early physiologists, and there is evidence to support both of these, as well as evidence to refute both of these as well. So these are just some of the theories as to how enzymes behave, although no one has ever really identified or visualized this and kind of proved it definitely. So the first one is the lock and key model. This is great at explaining the way that an enzyme binds and the way that the binding is specific, this kind of lock and key fit. So here is our enzyme. Here is the active site. The active site is wherever the substrate or product would bind. And so the lock and key model says that the enzyme has a specific fit, kind of like a key and a lock, to bind, and then that will generate the product on the other end. So in this instance, only substrate C would be able to bind to this enzyme because it's the only one that would fit. Again, this is great at explaining the specificity of enzyme activity, right? We know that this is true in our body, that we have multiple enzymes, but they only bind to unique substrates. But what it doesn't explain, right, what kind of refutes this theory, is that it doesn't explain the reversibility of most enzymatic reactions. The fact that the substrate can bind with a specific fit, but then how does the product also bind with that same specific fit? So the problems or the issues with this theory is that it doesn't explain the reversibility of what we know exists with enzyme reactions. The second model is the induced fit model. And the induced fit model says that the substrate will get in close proximity to the enzyme and it will induce a fit. It will sort of result in a configurational change of the enzyme such that it is going to fit perfectly in that active site. And then we have the product being produced on this side of the equation. Likewise, if you wanted this to proceed in the reverse direction, the product would get in close proximity of the enzyme once again, and it would likewise induce a fit such that the substrate can be produced on the other end. So this theory is better than the first one in terms of it explains the reversibility, right? If something gets close enough, it sort of configures it to be a perfect fit, and then it can proceed in both directions. However, the problem with the induced fit model is that theoretically, any substrate can induce a fit to any enzyme, right? So there are holes that can be poked in both of these theories, but it's important to understand both of them, how they differ, and how they can be supported, how they do explain certain aspects of enzyme function. So the lock and key model explains the specific fit, and the induced fit model explains the reversibility. Let's think about some other important elements to enzyme functioning. These are cofactors and coenzymes. Cofactors are going to be non-protein components that some enzymes require in order for them to sort of just stay structurally together, right, to hold their functional conformation. Some of the examples here are some of our trace metals, so magnesium, copper, zinc, right? Those are examples of components or cofactors that are attached to the enzymes that are going to help the enzymes carry out their role and actually help the enzyme carry out the reaction. Okay. Now, coenzymes are going to be proteins, first of all. This is where cofactors differ. Cofactors are not proteins, they're trace metals typically. Coenzymes are certain proteins they can be vitamins in some instances where they have no catalytic activity. They do not help the reaction proceed as such, but their function is sort of a supportive role to the enzyme's function. Again, this can be vitamins, B12 is an important coenzyme in some of our reactions, and they can be small chemical groups like phosphate can be transferred by way of some of these coenzymes. So I want to give an analogy that I like to give here to help us describe and distinguish these two. A coenzyme is like the steering wheel in a car, okay? It's nice to have, but it's not going to move the car. It's just a supportive role in the car. The cofactor is going to be like you in the car, okay? The cofactor is an integral part in the driving of the car, okay? Whereas the coenzyme, the car could essentially be moving, right? But if you're not in there, it's not going to be functional. So the cofactor is more integral, like you in this passenger seat. The coenzyme is really just a supportive role that's nice to have in terms of the overall function. Okay, that may be a terrible analogy, but hopefully you get the distinction between the two. Let's think about enzymatic rates and factors that affect how fast or slow a reaction can proceed. Now, things that are affecting a reaction rate would be the catalytic weight of an enzyme individually. This can sometimes be determined by how many molecules or how many product or substrate is generated in unit time. So how fast is that enzyme at making product or substrate? The second factor is the substrate concentration. How much substrate exists? How much substrate is available for the reaction to proceed will certainly determine the rate. The enzyme concentration, similarly, how much enzyme exists, right? Fourth is the affinity of the enzyme for the substrate, whether or not there's a strong affinity or attraction versus whether or not there's sort of a repelling between the enzyme and its substrate. Third is the temperature and pH. Now, this is one that it can increase the rate of the reaction. If we have an increased temperature, naturally, molecules have more thermic energy and they're going to be moving faster. And so the reaction could proceed faster to a point. If the temperature exceeds a certain threshold, we're going to denature the enzyme. So the temperature and pH can increase the thermic energy, right? The energy in particles, which can help the reaction speed up. However, that is only to a certain threshold. Beyond that, because enzymes are proteins, we can eventually denature these proteins in the process.